Hello, this is Miss Dagenford, and we're going to start our translation notes with just some introduction to some of the key features and key players involved, the molecules that are working. So remember the central dogma. The central dogma is the process by which a protein is made. We're going to start with a gene, and we're going to transcribe that gene into an RNA product. Now, you're used to hearing mRNA, and of course, mRNA is one of the products, but we can also transcribe DNA into a transfer RNA. And so this would be a strip of RNA, and because it's made with a certain sequence, it will fold back on itself. And so notice um, if I zoom in on this, you can see that there are complementary base pairs on this strand. And what they do then is fold back and create hydrogen bonds. And these hydrogen bonds will allow this very specific shape to be created. Now, what you're seeing here is simply a schematic drawing, more like if we laid out a piece of origami and laid it flat on a table, that's what it would look like. But when it folds up, it has a slightly different shape to it, and we'll look at that in a second. On one end of the transfer RNA, the amino acid will connect. And so this is the amino acid. And then on the other end, there are three nucleotides that will match and complementary base pair with a codon. And we call this area the anticodon. So it is going to be complementary to a codon. Now, as it folds and twists, this is what it actually looks like. You can see here all of the hydrogen bonds that are causing this very unique shape. So it kind of has this upside down L look. So we're seeing different models here. We see one that's just more of a outline of what it looks like with all of the loops and turns. We see an actual picture, but we also see then just a kind of a cartoonish or a symbolic view of a tRNA molecule. So again, here's where the amino acid would connect. Here's where we'd have the anticodon down here. And then this would be the codon that it matches. Now, the tRNA, even though it's technically transfer RNA, I sometimes like to think of it as translate. Because that, to some degree, is what it does. It has two languages that it kind of reads, if you will. It connects to RNA, mRNA, but it also connects to an amino acid. So here's kind of the bridge between these two systems. The other player that we have is a ribosome, and we've talked about ribosomes before. They make protein, and we know proteins are made of amino acids. So here's where we're kind of putting everything together. We've got a ribosome that is made of ribosomal RNA, and then also some protein itself. And so think about big strands. There we go. Think about a big strand of RNA, and it folds and curls and folds and curls back on itself. And as it does so, it kind of creates this superstructure. And then in the midst of it, you also have proteins that are kind of bonding together with the RNA. And so that's kind of what we're making here is a large subunit and then a small subunit of a ribosome. And on the large subunit, you have three areas, and they're designated E, P, and A for exit, the peptidyl, or we can think of peptide, and amino site or A site. Now, you can either think EPA, or if you want to think ape backwards, I've had students like that too. And so the small subunit is going to be the one that binds to mRNA first. So this is This is initiation of our translation. We're going to have our small subunit bind with the mRNA. We have our first tRNA binding to a start codon with the appropriate amino acid. And then we have the large subunit coming in, and this requires ATP for this step to happen. And once we have everything started, then we start a cycle of the elongation. And with the elongation, I like this diagram. What you want to add in your notes, you want to add some codons and specific, and I just chose CCC and UAC, and you'll see why, because what you want to notice is what the ribosome does as it continues on. So here are our steps. We're going to start at the top. Now notice we're kind of mid-translation. We've got 1, 2, 3, 4. Let me go ahead and number them. Actually, 1, 2, 3, 4 amino acids. And we've got a free area in our ribosome. That's going to be the A site. We know that because it's got one amino acid, and it's going to come and complementary base pair with our codon. And so a new tRNA arrives at the A site. Now, again, it requires energy, and what's going to happen is this ribosome is going to act like an enzyme. It can catalyze a new peptide bond between the new amino acid and the existing polypeptide that's already there. So again, notice we've got our codon, CCC, the next codon, UAC, and from those, you should be able to tell me what the anticodon is for this one, and the anticodon for that one. And just to go ahead and answer that, you should have said that the anticodon is GGG and AU, because we're still talking about RNA. It's not DNA. AUG. And so that's the anticodon. Now, if you were to use the chart to find your amino acid, make sure you remember that we're going to use the codon chart, not an anticodon chart. You're always using the codon for that. Okay, so once this peptide bond has been made, this whole system can shift down the mRNA strand. So we're going to see the ribosome move to the right, or you can think of the mRNA moving to the left. And so watch what happens to our codons here, and watch where they're going to be, because the next couple of steps are going to show this translocation. So we're going to break this bond that's holding that circle amino acid. We're going to break that bond, and as that happens, it's going to translocate. It's going to move down. Now notice CCC is in the empty spot, the exit site, and there goes our tRNA leaving. The UAC is now in the P spot, or the P site, and the A site is now empty. And so notice the position of our diamond or square. It's now in the P site. And we could start over, but of course, if we were to draw this, we would need to draw the ribosome like this in its new spot. That's the only, I wish we could show, it would be moving down. But what then happens is it just keeps cycling down and cycling down. And so you end up with the whole mRNA strand being red. Now, I believe that gets us to the next video, so I will stop there.